Thanks for that introduction. And so, as Jill said, I'm going to be talking today about the formalism of polyphasic linkage and the impact of ligand binding on the formation and dissolution of biomolecular condensates. And so this work was done in the lab of Roa Papu in collaboration with Furkan Dar and Anand Posey. And so spatial or biomolecular condensates can spatially and temporally organize cellular matter in the cell. And so these are various condensates that have been shown to be formed in eukaryotic cells. And so condensates seem to form via phase separation, where you get interactions among multivalent molecules known as scaffolds. So here's an example of DHH1, which is a multivalent molecule that is a scaffold of the P bodies, and you see these foci in these yeast cells. And so the nice thing about this is that we can think about these condensates in the terms of phase separation and phase transitions. So particularly phase separation is a density transition in which above a particular scaffold concentration, you get this coexistence of the two phases, a protein dilute phase and a protein dense phase. And so what this suggests is that we can use this quantitative lens of phase transitions and phase separation in order to understand the formation and dissolution of these condensates, but also the regulation of these condensates. And so if condensate formation is driven by a single scaffold, the phase behavior can be described as a function of the scaffold concentration as well as the interaction strength, which can be modulated with things such as temperature or salt or pH. And so what you have is a binodal, which denotes the region where phase separation is realized, and you have this coexistence of those two phases. So for a particular temperature outside of this regime, you'd see a single phase. However, once you cross the critical concentration, which we're going to denote as C del, you see this increase, or you see the coexistence of the dilute phase and the dense phase, where it's given, the dilute phase concentration is given by C del, and the dense phase concentration is given by C dense. And so what we wanted to think about is how do cells actually exert a thermodynamic control over phase transitions driven by scaffolds? And so you can see here that you could just change the scaffold concentration, or you could change things such as temperature, but the cells don't really change that much in terms of temperature. And so are there other ways that the cells actually regulate the formation and dissolution of condensates? And so if we look at what has been done in terms of compositionally profiling these condensates, what it seems to suggest is that these condensates actually contain tens to hundreds of distinct types of macromolecules, but only a handful actually appeared to be important for driving scaffold or condensate formation. And so this suggests that a lot of the other molecules are going to be regulatory molecules within these condensates. And so here we're defining ligands as molecules that preferentially bind scaffold molecules across the phase boundary, leading to regulation of phase separation. And so Wyman and Gill introduced this idea or formalism of polyphasic linkage, which actually describes the linkage between ligand binding and the modulation of phase separation. And it's in its most sort of simple form, it's given by this equation here, where we have the saturation concentration of the scaffold in the presence of the ligand equals the saturation concentration of the scaffold in the absence of the ligand times the ratio of the binding polynomials, which in both phases. And this is true if the ligand can actually bind both phases. And so what this suggests is that the scaffold itself without any ligands will have a given saturation concentration. However, if the ligand preferentially binds the dilute phase, the binding polynomial for the dilute phase will be greater than that of the dense phase, and you'll see an increase in your saturation concentration. And if the ligand preferentially binds the scaffold in the dense phase, you'll see the opposite, and thus you'll see a decrease in your saturation concentration. And so what this suggests is that if we knew the underlying binding polynomials within both phases, we could directly get out what the saturation concentration of the scaffold should be in the presence of the ligand for various concentrations. And so in its simplest form, where we say that the scaffold has independent ligand binding sites, the binding polynomial would be given as the followed, where you have the cumulative association constant and your free ligand concentration. However, in terms of actually extracting binding polynomials, there's actually several challenges. One, a lot of these molecules that form condensates actually can form at very low concentrations, making it hard to actually even examine the dilute phase. Additionally, when you're doing measurements in the dense phase, that can be a convolution of various interactions, and so that can be hard to extract your actual binding affinities and things like that. And then thirdly, the scaffold binding sites are not always well defined, particularly because a lot of these molecules are intrinsically disordered and have a lot of low affinity binding sites. So instead, what we want to do is we want to take this formalism, but then use it to try to understand sort of what features actually promote or suppress phase separation. And so for this, we're turning to coarse grained models so we can design various ligands and see the effect on the saturation concentration in the presence of ligands. And so the idea would be that given a whole set of molecules that are associated with condensates, we could potentially use this to predictively classify these various molecules, as well as rationally design ligands. And so this is similar to the work that's been going on there at Cambridge as well in the Corey Parter lab. And so we're looking at kind of finding similar rules. So the rest of my talk is focused on answering sort of three general questions. The first is what features of ligands actually allow for precise control over homotypic scaffold phase behavior? And then sort of the sub questions there are how do we design ligands that actually promote or suppress scaffold phase separation? And do the features that change when phase separation is driven by multiple scaffolds, which might be more of the case for these biomolecular condensates that we're seeing? And then secondly, I wanted to ask, how do we experimentally assess a ligand's modulatory effect on scaffold phase behavior? And given that partition coefficients have been readily calculated, are they helpful in this case? And then third, can we experimentally modulate phase behavior of scaffolds using ligands? And then here I'm gonna show an example that we used in the lab using the Huntington protein. So to answer the first set of questions, what we turned to was a polymer simulation using a lattice engine where we have a polymer of stickers and spacers. Here, the sticker sites are the regions that are going to drive phase separation through their crosslinks, and then the spacer sites are the ones that are sort of regulating. So here we have 5 explicit stickers and 2 explicit spacer sites. And then we're looking at the regulation of the scaffold phase behavior using 5 different types of ligands. We have 2 monovalent ligands, one that interacts with scaffold stickers and one that interacts with scaffold spacers. And then 3 divalent ligands, one that interacts with scaffold stickers, one that interacts with scaffold spacers, and one that interacts with scaffold stickers and spacers. And so, as I kind of said before, what we're interested in doing is really focusing on a particular temperature, since that's not really changing much in the cell, and then actually seeing what happens if we change the ligand concentration. And so if the saturation concentration decreases, that would mean that it would promote phase separation. If the saturation concentration increases, that means that the ligand is destabilizing phase separation. And if it's greater than the initial scaffold concentration, that means we don't see any sort of phase separation. And so what we find is for the monovalent ligand that interacts with scaffold stickers, we see a destabilization and pretty much a complete abolishment upon ligand binding. And so the way that this works is that we're getting competitive binding with those scaffold sticker sites, and we're getting no supplementary cross-linking because it's a monovalent ligand. Interestingly, when we have a monovalent ligand that interacts with scaffold spacers, we also see destabilization and eventually abolishment as you increase the ligand concentration. Here, again, we don't have any supplementary cross-linking, but instead of direct interactions that are blocking those sticker sites, what we're getting is an increase in the excluded volume of the spacer region, which changes the networking ability of those sticker interactions. And then third, when we look at the divalent ligand that interacts with scaffold stickers, what we find is destabilization and eventually abolishment. Here, again, we're getting competitive binding with those sticker sites. And although we're getting supplemental crosslinks, it's not enough to compensate for the loss that you're getting for the higher valence of those interactions within the scaffold. However, when we go to a divalent ligand that interacts with scaffold spacers, now what we see is the promotion of phase separation. And this is due to the fact that you can maintain those scaffold sticker-sticker interactions, but you can also have these supplementary and complementary crosslinks through those spacer regions. And then lastly, when we have a ligand that binds both stickers and spacers, when all the interactions are equivalent, we see a weak promotion of phase separation. But what's interesting about this ligand is that depending on the relative strength of the interactions for both the sticker-sticker interactions, but also the interactions between the ligand and the scaffold, is that you can get a whole range of modulating ability from greatly promoting phase separation to greatly destabilizing phase separation. And so what this suggests is that the valency, interaction type, and interaction strength is going to determine the effect on your saturation concentration, particularly monovalent ligands, as well as low valent ligands that explicitly bind stickers are going to destabilize phase separation, whereas these ligands that can bind the spacers and are multivalent have the ability to potentially promote phase separation. And so this is looking at the ability to sort of dissolve or form condensates, but that's not the only thing that may matter for function of condensates in the cell, particularly the underlying structure and the concentration in the dense phase is also likely important for function. And so we also took a look at the effect of these different ligands on the organization of the dense phase. Where here we're taking the concentration of the dense phase in the presence of the ligand, comparing that to the dense phase of the scaffold in the absence of the ligand, and similarly for the dilute phase. And so for our ligands that destabilized phase separation in terms of the dilute phase, what we see is also a decrease in the saturation concentration in the dense phase. So we're getting this shrinking of the binodal, where here the circle size is denoted the ligand concentration. So the larger the circle size, the higher the ligand concentration. So as you get larger and larger ligand concentrations, you further and further destabilize the dense phase. Additionally, if we look at the divalent ligand that interacts with scaffold spacers, we don't see much change in the dense phase. The scaffold concentration is relatively maintained. However, when we look at the ligand that actually didn't change the dilute phase concentration much, it can actually have a relatively large effect on the dense phase. And so what this suggests is that even ligands that don't really change the saturation concentration in terms of the dilute phase, it can have effects on the underlying structure and organization of the dense phase, which may contribute to sort of the function of the condensate. And just to interrogate this ligand a little bit more, we can look at sort of the number density or radiomolecularity profiles, where we can compare that to in the absence of the ligand, which is the gray profile here. And what we see is that the sticker-sticker number of interactions doesn't really change. However, it shifts to the right, suggesting that we're increasing the correlation between stickers, and that's actually why we're getting this dilution of the scaffold in the dense phase. And so what this suggests in terms of homotypic phase separation is that ligands can be used to regulate the formation and dissolution of scaffold-driven phase separation. If you have ligands that provide additional networking interactions, you can promote phase separation. Whereas if you have ligands that disrupt networking interactions but can't compensate as well, then you will also suppress phase separation. And it's also important to remember or to realize that ligands can tune the compositional profile of the dense phase, and this can have effects on function. And so this was all shown for a single scaffold driving phase separation, but the question that continues is sort of what happens when you have multiple scaffolds that are driving phase separation, which is the case for a lot of these biomolecular condensates, where heterotypic interactions are actually going to be important for driving phase separation. And so when phase separation is driven by interactions on multiple scaffolds, the phase boundary is now constructed by a hyperplane of these concentrations of all relevant scaffolds. So here I'm showing a schematic where we have two scaffolds that are interacting via purely heterotypic interactions, and we see this closed loop kind of phase behavior. Now, the actual shape of the phase diagram is going to give you information about whether homotypic or heterotypic interactions are in play. And so, although we've looked at a little bit of both, today I'm just going to show you an example of a coarse grain model where we looked at heterotypic interactions, where we have two scaffolds, scaffold A and scaffold B, both of valence 7, and a ligand that is similar to scaffold A, but it's of valence 2. And we want to see the effect of that ligand on the scaffold phase behavior. And so we see this closed loop behavior, but what's interesting is that we find that the low valence ligand can actually suppress or promote scaffold phase behavior depending on the relative concentrations of the two scaffolds. So when scaffold A concentration is greater than scaffold B in this regime, we actually see the suppression of phase separation. This is due to sort of the sequestration of scaffold B away from those scaffold-scaffold interactions. In contrast, when scaffold A concentration is greater than scaffold, or scaffold B's concentration is greater than scaffold A, we actually see the promotion of phase separation, where the ligand can actually contribute to networking interactions and promote phase separation. Additionally, we see that these ligands can destabilize or reduce the concentration of the scaffolds in the dense phase. And so overall, what this suggests is that having ligands that change the networking interactions will either promote or suppress scaffold phase behavior, where if you can add additional networking interactions, you'll promote phase separation. And this is true whether sort of the phase separation is driven by a single scaffold or multiple scaffolds. However, when you have multiple scaffolds, you actually need to know where you are in the phase diagram because the relative total concentrations will determine how the ligand actually regulates the overall phase behavior. So the second question that we wanted to ask was, how do we experimentally assess a ligand's modulatory effect on scaffold phase behavior? And particularly, are partition coefficients helpful? So partition coefficients are defined as the ratio of the concentration of the molecules in the dense phase divided by the dilute phase. And the reason why we're interested in this is that when these condensates have been compositionally profiled, a lot of partition coefficients have been generated. And so this is a case for stress granules and p-bodies, where only these G3BP1 or BP2 are actually the scaffolds in this case. And so a lot of these other molecules are likely to be regulatory molecules. So is there information from these measurements that have already been done that tell you if these molecules are sort of promoters or suppressors of phase separation? So to answer this question, we went back to our initial homotypic phase separation model, and we looked at the correlation between ligand partition coefficients and the change in the saturation concentration of the scaffold. And what we find is not a great correlation, where we have cases where ligands that actually suppress phase separation can actually have a higher ligand partition coefficient than those that promote. And just for comparison, which something that should be directly correlated, which is the scaffold partition coefficient, we do see a nice correlation. And so if we plot this in a different way, where we sort the ligand partition coefficients, and then we color the bars by their effect on phase behavior, again, we see that those that suppress can have a larger partition coefficient than those that promote. And it's really the most strongly interacting ligands that have the highest partition coefficients, suggesting that strongly interacting ligands, regardless of their effect on scaffold phase behavior, will have large partition coefficients. And so what this suggests is that these ligand partition coefficients are actually a convolution of various factors, including the binding strength, but as well as the ligand concentration, because ligand partition coefficients as well, once you get to very high ligand concentrations, the partition coefficients will go to 1. And so unfortunately, we're not going to be able to understand their regulatory behavior just by looking at these partition coefficients. And so this seems to be consistent with what was shown by Sanders et al. In 2020, where here we're looking at caprin-1 and USB10, which are two regulators in the stress granules. They have similar partition coefficients, but they showed that they actually have drastically different effects on stress granule formation. USB10 is actually a suppressor, whereas caprin-10 is a promoter of phase separation. And so what we need to be able to do is go back and take a lot of these molecules that are associated with these condensates, change their concentration, and actually measure just the scaffold concentration to see how it's changing and whether these molecules are promoters or suppressors of phase separation. And then lastly, we wanted to look at sort of an example work if we can experimentally actually shift these phase boundaries. And so Huntington is a protein that is associated with Huntington's disease when its N-terminal polyglutamine stretch gets expanded. And it's been shown that profilin, when overexpressed in cell models and fly models, it can actually suppress Huntington phase separation and toxicity. And so we hypothesized that profilin might be working as a ligand and shifting the phase boundaries of Huntington. And so Ammon Posey in the lab did in vitro studies where he took an expanded polyglutamine Huntington construct and profilin, and he increased the profilin and showed that we see this increase in the saturation concentration, suggesting you're destabilizing phase separation. And so this suggests that this destabilization that we see in vitro then might have effects in cell, particularly in reducing potentially toxicity, but also suggests that just being able to change those phase boundaries might affect function of sort of naturally occurring condensates as well. And then we were able to do quantitative binding assays as well as coarse grain simulations and atomistic simulations to show sort of how these interactions might be happening, where it seems that profilin is binding a spacer region within Huntington and then weakly binding the sticker region as well, leading to this destabilization of networking interactions through the polyglutamine region, which is sort of the sticker within Huntington, through both competitive and non-competitive excluded volume effects. And so what this suggests is that we can use this formalism to modulate the phase behavior of scaffolds using ligands, and that combining sort of these quantitative experiments with simulations can yield additional information for exactly sort of what interactions are leading to these changes in the phase behavior. Okay, and so just to wrap up, I just wanted to leave you with that these polyphasic linkage concepts provide insights for both understanding natural regulation as well as pharmaceutical applications. So in terms of natural ligands, when you change sort of their concentration, you can see the effects on the scaffold phase behavior. But what's interesting is that in these condensates, you have a whole network of ligands, and particularly as has been shown in the stress granule case, you have ligands that both promote and suppress phase separation. So the question is, what is the push-pull effect? Why do certain ligands win out? Why do you see the formation of these condensates when you have both? And so where we want to take this is to start to look at theories and models of networks of ligands, where then we can go back to sort of the underlying equations and binding polynomials and think about a series of various ligands and what it takes for certain ligands to win out within a whole network of interactions. Additionally, these ideas have pharmaceutical applications where you could screen a whole set of various ligands and see their effect on scaffold phase behavior. And then additionally, the rules that we've extracted here, as well as others, including people listening to this talk, we can use to have rational design of ligands if we have some information about the stickers and spacers within the scaffold themselves. And so with that, I'd just like to thank my lab and thank you for your attention.